this course, he will talk about this will be the first presentation about the endocannabinoid. So Andreas, you have remember you have if you want to interrupt him, you can do it. This is a very dynamic course that you can interrupt whenever you want. Yes.
One question will ask. I'm going to have just the video recording.
Yeah, no, they are just PDF file, no, because I mean, there will be some unpublished data. Of course, public data cannot be on the PDF file. This is the advantage of this course. Most of the data are still unpublished. Some data at least are still unpublished.
Okay, so that's their AV guy.
Oh, really?
Yeah.
Hello, can you hear me? Can you hear me?
Yes.
Works. Okay. Thanks for having me here. It's a pleasure to present some of our the research topic of my laboratory. We are interested in the endocannabinoid system. And I'm just having some technical problems here. Yeah. You're probably familiar with these substances here. This is hashish, or this is cannabis oil spread on a cigarette. I'm not showing a picture here of marijuana, but these are the most commonly used illegal drugs worldwide. The signaling system that we're working on actually kind of was discovered when people were looking for the active ingredient. This is just a cartoon of the marijuana plant, cannabis sativa, that lent its name to the whole system. And the whole field was really started when people were isolating or succeeded to isolate the active ingredient in marijuana. And that was exactly 50 years ago. By this person, Rafi Michulam, at this time he was at the Weizmann Institute in Tel Aviv. He is now Professor Emeritus at the Hebrew University in Jerusalem. And this is the active structure of or the structure of the active ingredient in cannabis. It's called delta-9 tetrahydrocannabinol, or THC, as we typically name it. And you can see it's a lipophilic substance. I'll tell you later that it was for a long time not known how this substance acts in the brain. So, as I mentioned, this is the most commonly used illegal drug worldwide. And you can see actually this is a relatively recent survey from the United States from teenagers. You can see that 8th graders already had a 15% lifetime experience, and it increases to 42% in 12th grade, and I don't know what the number is among college students, but then it's very high. So a very, very large percentage of all teenagers in the United States gets contact with this drug. And same thing in Germany. So the lifetime prevalence of cannabis use has steadily grown over some period of time, and now it seems to be relatively stable. So this is a drug that is very frequently used, especially among youngsters. And you can see here the survey already starts with people at 12 years or younger. And I want you to keep this in mind. This is one trend that we're observing. The other trend that we're observing is that the cannabis material increases to have a higher and higher THC content. So in the early 90s or late 80s, actually, you can see that the typical cannabis THC content in this cannabis preparation was well below 5%. And now it has exceeded actually 10%. So the cannabis that we're seeing today is much more potent than the cannabis that we're seeing in the past. And this may have an effect, actually. Several studies indicating now that actually that even though the stability of the overall prevalence has remained constant, there seem to be more and more problems related to the use of cannabis. Cannabis use disorders, and they have just been much more reported than this 10 years ago. So cannabis with high THC content is prevalently used among teenage children, among very young teenage children, and this this has been actually correlated to an increasing number of problems. So this is almost all that I want to tell you today about cannabis use and abuse, and I want to dig a little bit now into the biology and the effects of cannabis that it may have on individuals at certain ages.
Can I have a quick question?
Yeah.
What happened in 2004 in Germany? Because we see a decrease in, starting from 2004 in marijuana use. Is it related to the increase in content?
Yeah. I'm not sure. You mean this year, right?
Yeah.
Why is it dropping all of a sudden by a few percent?
Especially in 12 to 17 years.
Yeah.
That's happening all around Europe. In Spain, we have exactly the same profile of the consumption. If you look the numbers in Spain, in 2004, we had even a higher percentage of use than you. We were the leaders in consumption in Europe in 2004, and now it was not the case. And in 2004, there was a decrease. And the decrease in our case, but in our case, was mainly for the risk perception started to move a little bit. You know, the perception of risk of consumption started to move a little bit, and also the amount of drug that was available was just at the top. So there was a lot of promotion of cannabis consumption at the beginning of 2000 that just increased a lot the consumption. In 2004, at the beginning of 2000, there was campaigns just explaining what cannabis is in Spain, so the perception of risk increased a little bit. The amount of cannabis increased a lot, but remains the same in 2004, and just started to decrease. And this decrease, at least in Spain, was associated to a very big increase in consumption in alcohol. I don't know in Germany, but you see the decrease in cannabis and the amount of alcohol that is consumed just for binge alcohol consumption, just for let's say intoxication, no? Was increased. So it was just in the amount of drug consumers was the same, but more alcohol, less cannabis from 2004 in Spain. No.
I think the economy also was affected on that time because
Sure.
the price of the cannabis in 2004 started to increase. It was very cheap at that time, but again I started with it. So the people just say, okay, I'm gonna be high for just a few hours with the cannabis, but if I drink alcohol, I can keep it with a cheaper price for a longer time.
But at least in Spain,
Alcohol also dropped.
it was also up in Germany.
I'm not sure actually whether the binge consumption dropped after 2004. I don't really know. I was wondering whether could be related actually to the anti-smoking campaigns that became very active in Germany. The anti-smoking campaigns. So, I mean, I think the law was roughly implemented in 2004 that banned actually smoking from public places. So I'm not sure whether this contributed or not.
In Spain, the campaigns against tobacco are a bit before, and the dropping in tobacco consumption was really much strong than in the case of cannabis, but for sure cannabis an influence. But now, what we also observe in Spain is that the perception of risk is completely independent, tobacco and cannabis, because cannabis has now increased a little bit the perception of risk. The perception of risk of tobacco consumption is dramatically increased, what is much better in order to decrease the consumption, of course.
Yeah, but at least in Germany, people always smoked also marijuana in pubs and bars. And so since nobody's smoking anymore, people, I don't know. So I don't really know. Could be education, could be awareness actually of the risk, could be economic factors, maybe a multitude of different factors. So let's talk a little bit about the biology of cannabis. I showed you the structure of THC a few minutes ago, and you know this is a very lipophilic structure. And because of the structure, people for a long time assumed actually, or were not sure whether this would act via attaching to a specific protein, or whether it would just intercalate into the membranes of neurons and alter the biophysical properties of neurons. And that was discussed for quite a while, and the solution to this problem actually was only found when high affinity radioligands became available. So then people started to synthesize structural analogs to HC, which were able to compete with THC for specific binding sites in the brain. And these compounds could be used then actually to label the sites to which THC would bind in the brain. And this is showing you such a picture here. It's a very famous compound, CP55940, that was used. It's a picture from Miles Herkenham. And there were stereoisomers for this particular compound. And people found that only one of the two stereoisomers showed this binding pattern. And this isomer specificity is usually taken as an indicator for protein to which the substance binds. Will it just intercalate into the membrane? Both stereoisomers would do this. So this was evidence for a specific protein present in the brain to which THC combined, and this binding site actually replaced if you expose it to high concentrations of THC. So one thing you can see from this picture, actually, it's binding sites are very abundantly present throughout the brain, particular high concentrations in basal ganglia structures here, also in the hippocampus, also in the cerebellum. Generally, we have binding throughout the entire brain. And now I can tell you a little personal story. At that time, in 1989, I was looking for a postdoc position in the US, and I went to a lab at NIH, and there was a young postdoc, Lisa Matsuda, working there. and she was working on a G protein coupled receptor. So she cloned the G protein coupled receptor, but this was an orphan receptor, so no ligand was known. And her task was to find the ligand for this receptor, de-orphanizing this receptor. And this is very tricky, actually. It is still tricky today. We have still a relatively high number of orphan receptors. It was particularly tricky 15 years ago. So I thought this is a really stupid project to do. And then I went home and finally decided to go to NIH. And 6 months later, I visited with Lisa and asked her, how's the project going? She said, oh, I discovered the cannabinoid receptor. So much to the stupid project. And what happened was actually that Lisa did an in situ hybridization to look for receptor expression in the brain. And she got a picture from the in situ hybridization, and Miles Herkenham, who produced that picture, was in the same lab. And he happened to have this picture presented on the outside of his door. So one day Lisa actually walks by, literally looked at this picture, and thought, well, this looks very similar to my in situ hybridization. Maybe it's the cannabis receptor. And when she tested it in tissue culture, indeed it was, and it was published also in 1990. And now we know we have actually two cannabis receptor. This one, the brain receptor, we call it CB1. There's another one that was cloned, I think, just 2 years later by Monroe and colleagues. This receptor is mostly expressed in peripheral tissues, very prominently in immune cells, but it's also found in bone. I'll show you some evidence also later. And there's currently a very active discussion in the field whether CB2 receptors are also present in the brain or not. I believe in the meantime it is present in a few cells in the brain, but I have been a disbeliever before. But most of the effects on the CNS actually mediated by CB1 receptors. So, what are the endogenous ligands? The endogenous ligands were discovered at about the same time, actually, or shortly after the receptors were cloned. This is the first one that was cloned, ethanol, we call it also anandamide. This was isolated by Bill Devane, again in Rafi Michulam's lab, the person who also discovered THC. You can see it's in arachidonic acid derivatives. Another substance was discovered not much later. It's 2-alpha-glycerol or 2-AG. And both of these endogenous ligands bind very prominently or bind and activate both receptors. 2-AG is present at much higher concentrations in the brain than anandamide. So, while I was at NIH, we then actually started to address the function of the endocannabinoid system using transgenic mouse models. What we wanted to do is produce mice that lack these two receptors, and then we wanted to see, first of all, what are these receptors doing, and which effects of THC and other substances are mediated by these receptors. So we made knockout mice by deleting basically the entire receptor, and that just shows you a picture with kind of proof of concept. So this picture you're already familiar with. These are the binding sites in wild-type mice, and if we delete binding sites for CP55 and I40, exactly the same substance, radioligand I showed you before, and when we delete CB1 receptors, there is no residual binding of CB1 receptors in the brain. So to us, this was already a very strong indication that most of the effects, if not all of them actually, of THC on the brain should be mediated by CB1 receptors. Now, because really all binding sites are gone, it was for me, or this was an argument for me actually, that CB2 receptors can only play a minor role. However, there could be CB2 receptors still present, but the binding, if they are present in the brain, that's what's being discussed, the levels are so low actually that we cannot detect them with normal binding using this methodology. Now, if we knock out the receptor, then animals basically do not seem to show any responses to THC at all. So if you inject THC in wild-type animals, these animals assume a very, very typical kind of stretched position here with an extended tail, and they remain, if the dose is high enough, completely motionless for a very extended period of time. And if you inject the same dose in CB1 knockout mice, they're not bothered actually by this at all. They walk around normally. They still don't like to be photographed, I think, but they don't really respond in any visual manner to THC. So how do we measure the effects of THC in animals? Billy Martin from the University of Richmond has devised a so-called tetrade of assays. And the tetrade of assays is based on 4 prominent behavioral responses that one can observe to cannabinoids. One is THC, produces analgesia, inhibits pain responses. Wrong picture here. Another one, it produces a drop in body temperature. It produces catalepsy. Catalepsy is when an animal remains motionless in an unnatural position. If you put it on a ring, a normal mouse would just jump down, a cataleptic mouse will remain there. If you put a mouse on an inverted platform or one way, put the head down like this, a normal mouse will just move around. And the fourth thing, this is where I meant it, wrong picture, it also reduces open field activity of the mice, which we can measure in an open field. Okay, so this is the tetrade of assays that's typically used to investigate THC effects. And if we do this, actually, you can see that for hypothermia, we see a profound drop in body temperature in wild-type animals. Those dependently, it goes up to down to by 5 to 6 degrees Celsius. We see a very, and this is completely gone in knockout mice. They show no drop in body temperature. Same is true for ring catalepsy. We see very profound catalepsy with THC, completely abolished in knockout mice. Tail-flick analgesia is still present in CB1 knockout mice. We don't see any hot plate analgesia. So we still have some residual analgesia. And we believe actually that these residual analgesic effects of THC in CB1 knockout mice can be mediated by CB2 receptors. So most of the effects of THC are gone, but some effects actually peripherally mediated could still be present in these animals. So now, after almost 2 decades or a decade and a half of research, we understand the function of the endocannabinoid system in the brain actually quite well. And let me explain actually how we think the endocannabinoid system works. Shown here is an excitatory glutamatergic synapse. This is the presynaptic terminal. This is the postsynaptic terminal. And you can see that there's glutamate released from the presynaptic terminal that acts on ionotropic receptors, AMPA and NMDA receptors. Now, under normal excitation condition, actually, there is the glutamate activates only these ionotropic receptors under high excitatory conditions. So when these free synapses here release a lot of glutamate, there will be some glutamate spill over. And this spillover will activate now metabotropic receptors that are not shown here in this picture. It will activate metabotropic glutamate receptor, which are located at perisynaptic sites, just outside of the synaptic cleft. And particularly mGlu-R5 seems to be formed here. And the activation of these perisynaptic metabotropic glutamate receptors will then stimulate the production of endocannabinoids postsynaptically from membrane phospholipids through stimulating the activity of enzymes that we call the acylglycerol lipases. So these endocannabinoids, 2-AG here, can then diffuse back retrogradely against the normal information flow and activate here presynaptically located CB1 receptors. And the CB1 receptors will then, the activation will then shut off basically further neurotransmitter release. So what we have here is a negative feedback system at excitatory synapses. Basically under conditions of strong activation, this feedback mechanism will be activated and it will prevent or decrease the further release of glutamate. Now, the situation at GABAergic inhibitory synapses is still less clear because there are no endocannabinoid synthesizing enzymes directly on the postsynaptic sites of GABAergic synapses. So it's not entirely clear actually, but these GABAergic synapses contain also CB1 receptors. It's not entirely clear actually how 2-AG or anandamide gets to these CB1 receptors, but it's possible that we have a heterosynaptic activation of these CB1 receptors, but other mechanisms are also possible. Okay. One thing I wanted to mention is that endocannabinoids, of course, or cannabis is of course consumed to increase euphoria and all the effects actually through the activation of CB1 receptors. However, other drugs of abuse lead probably to a stimulation of endocannabinoid synthesis, activation of the CB1 receptors, and the system seems to modulate the activity of other drugs of abuse. And I just want to show you one example here, and that is alcohol. It has been observed actually that CB1 agonists stimulate ethanol intake and across different species. So these are ethanol preferring rats, and if you treat these rats, this is another synthetic antagonist, agonist with various doses of this agonist. You can see actually that the amount of alcohol that's consumed by these rats increases. You can also produce the opposite effect. Mice like beer, and they voluntarily self-administer actually beer, or they drink beer. And if you treat them with a CB1 receptor antagonist, the beer consumption goes down. For German, this is very sad. I'm just kidding. So indicating actually, and this is only this is specific for beer. It's not observed with near beer. Near beer is basically alcohol-free or very low, almost alcohol-free beer. So alcohol seems to be important actually for this effect of CB1.
Yeah.
So alcohol is quite caloric.
Excuse me. Mhm. Yeah.
Isn't it possible that CB1 actually regulates caloric intake,
Yeah.
therefore regulates alcohol?
Yeah, we know now actually that that CB1 modulates actually very much the consumption of many palatable substances, but there is probably also a specific effect of ethanol. So ethanol directly activates the reward system. You'll hear this tomorrow, I guess, from Rainer Spanagel. And this is probably enhanced by,
So it'll be less same for cocaine.
yeah, cocaine. Yeah.
Similar, we are going to discuss tomorrow.
Also?
But it's a very similar,
Yeah.
One question, the breaking point is similar for beer and for near beer.
That's interesting, isn't it?
Because I would expect a different breaking point.
Yeah, me too. This is not our study, but
I know.
you're absolutely right. Yeah.
Is the taste of beer and near beer the same?
The taste for beer and near beer is the same. Yeah, but still, the ethanol actually should produce some additional rewarding effect. And
Yeah, that's why I'm asking. So you might have a taste preference.
But in principle, you should have both the rewarding effect of ethanol and the rewarding effect of the taste and the caloric value of these drinks. It should be a higher rewarding effect in the case that you add alcohol, but it wasn't the case.
But if it's the first exposure, and they don't know.
I guess should not be the first exposure, because in the case of the first exposure, you never get alcohol self-administration. And Rainer Spanagel is going to talk tomorrow about that, but you're going to see that to get ethanol self-administration in animals, you need a repeated exposure, because if not, the animals, they don't like the bitter taste of alcohol. And you always must habituate the animals in order to get a voluntary self-administration.
I don't know what's happening here. Okay. So we actually also did some studies of this sort, and we collaborated with George Kunosh, who at that time was in Richmond. He's now the scientific director of NIAAA. To look for the effects of CB1 disruption on ethanol consumption. And one thing that George did was he first looked at ethanol consumption in old individuals. Basically, he was living in Richmond. He was living still in Bethesda and working in Richmond. So he came by to our lab in the morning to pick up mice, drive down to Richmond and do experiments there. And we first only had old mice for him. And so he did experiments with old mice, and there was basically no difference. If you look at ethanol preference here, there was very little difference between wild type animals and knockout animals. At least this was not significantly reduced. And this was very frustrating. And so George told us, you have to give us really young mice. Maybe your mice are too old. And so we gave him young mice. And now you can see actually that the young wild type animals, they drink much more alcohol than the old animals. And they drank significantly more than the CB1 knockouts. Yes, you have a question?
So for the old mice, how old were they?
It's a good question. I think they were about half a year old,
Okay.
5 to 6 months. But I would have to look up the exact number in the paper, yes?
Did you evaluate the CB1 expression in the wild type old mice?
Yeah, I'll come to this in a moment.
In
And also importantly, actually, when George treated the young mice with an antagonist for CB1 receptors, he could reduce alcohol consumption in these young animals. It's true for preference, it's also true for the overall alcohol intake. And this indicated to us actually that there is a difference in the way that old and young animals responded, and also a difference in the way that endocannabinoid signaling affected these responses in old and in young individuals. So this is not only observed for ethanol. You asked a question here whether this is specific for ethanol. No, it's also food intake is modulated by cannabis. We know this actually. If you smoke cannabis, usually you get hungry. And so food intake of starved animals is much lower in young CB1 knockouts, or also young wild-type mice that are treated with a CB1 receptor antagonist. And the CB1 receptor antagonist doesn't affect really food intake in old individuals. So there's a difference between young and old. Now there is not so much of a difference in the level of radioligand binding in the brain. There are differences, but the differences are small. Where people do see differences is in the signaling of the CB1 receptor. So if you do GTP gamma S binding assays from different brain regions, you can see that specifically in limbic forebrain structures, also the hippocampus, old animals show a reduced CB1 receptor signaling compared to young animals.
Then you don't see difference in the expression, only in the signaling.
It's yes, it's the signaling seems to be more affected by the age than receptor expression.
And this must be related because there is a deficit in the endocannabinoid ligands, yeah, I guess.
Oh, the ligand, the endogenous ligand also changes during aging. So we see lower 2AG levels in old individuals than in young individuals. But how this receptor coupling, the molecular mechanisms that are responsible for the reduced receptor coupling in old animals are not known in detail. So, but this alerted us to the fact actually that in the endocannabinoid signaling seems to change during aging, during the aging process. It peaks probably during, or it does peak during adolescence. During adolescence we have the higher binding, we have the highest GTP gamma S binding also, and we also find the highest level of 2AG. Then it declines, but throughout adulthood it remains relatively stable. There are also some studies in humans actually that all show that during adulthood it's relatively stable until at some still undefined period in old individuals it declines. So then if the individuals get older, then you should see really a decline in CB1 receptor levels, but also GTP gamma S binding and also endocannabinoid levels. So changes dynamically. And this prompted us actually to look for THC effects at different ages. Now, one thing that we were interested in was to look at very young individuals. For a simple reason, there have been many reports really showing that THC consumption during adolescence and puberty is a major risk factor for schizophrenia. So there's a very clear association. Kids that smoke cannabis have a higher risk of getting the schizophrenia later on. And that seems to be correlated with THC levels. For example, you see in South London, a higher risk actually attributed to a THC abuse than in North London. And in South London, people smoke very often cannabis oils, which have very high THC level. In North London, that's different. So this seems to be correlated. So what we did was actually we fed young mice with THC. We didn't want to inject them, so we delivered drops of milk to these young mice, which they voluntarily consumed and which contained THC. And we did it to during a very early period between P14 and P28. We also did studies at a later time point, but I'm showing you today only these here. Because this is a critical period, we think, during postnatal brain development, where there are many events happening actually that are modulated by cannabinoid signaling, or we thought cannabinoid signaling could be involved. For example, we have cortical pruning is going on, excessive synapses that are formed during postsynaptic, during postnatal development are removed, and we still have a very active myelination going on. So we wanted to interfere with these processes if they are CB1 dependent by giving them THC, and then we let the mice be THC free for about 4 weeks, and then we started with the behavioral testing of these animals. So if we feed mice, and this is an experiment shown with 28-day-old animals, so there's 4 weeks of age. You can see that actually when after they have eaten or licked up the cannabis milk, the activity of these animals is really going down. So we get substantial THC levels into these mice that show very, very clear behavioral effects.
What was the daily dose of THC?
We'll have to look it up. I think the daily dose of THC was in the order of 10 to 15 milligrams per kilogram. So it was, but only a single dose. It was a single relatively high dose. We use both, and I don't know which one is this here. So then, this is just to show you that THC produced an effect. We also measured THC levels in a few animals, and we indeed get blood levels that are similar to those that you would see for humans after smoking pot. So an animal experiment that a test that we did is called latent inhibition. Basically, this is a test. This is relevant to schizophrenia. It shows you basically a filtering system. In this test, you have what you do is you expose animals to acute tone first, so the animals become familiar with the tone, but the pre-exposure has no consequence for the animals. So just hear the tone over and over again. And they basically learn this tone is nothing, just there. But then actually we perform 3 tone shock pairings. So whenever we first give them a shock, tone, and then a small foot shock. The tone, small foot shock, tone, small foot shock. And now the animals should make a connection between the tone and the foot shock. And they usually do if we expose them just to random noise. But if the animals have already learned to kind of ignore the tone, this conditioning will be less effective. And that is an important filtering system for us actually that helps us to filter out signals that are important and discriminate these signals from those that are not important. And here the animals should have learned that actually the tone is not important, and then on the second day we just measure or evaluate the conditioned response. So pre-tone test, there's no difference between vehicle and cannabis treated animals. But then in the tone test, what you can see here is that the conditioned response, the freezing, that is an anxiety reaction of these mice that they show when exposed to the noise, is higher in those animals that were not pre-exposed to the tone, than in the animals that are pre-exposed to the tone. So here this latent inhibition is working normally. The animals have learned actually, if you want to disregard the tone, and they make the conditioned response is made less efficiently. If you look at the cannabis treated animals, we can very clearly see actually there's no latent inhibition. The freezing response is not different in the pre-exposed animals and the non-pre-exposed animals. So here the filtering system that is so important to learn to eliminate non-critical noises is disrupted by adolescent exposure to cannabis. And this is a filtering system that's also disrupted in schizophrenia patient. Another test that we did and that I'm showing you here is the disruption of social behavior. How do we, you know that schizophrenia patients, actually one of the hallmarks also reduced social contacts and affected social behavior. How do we test this? We put mice into a cage that contains 2 cages. One cage is empty and the other cage contains a mouse. So typically mice are more interested in the mouse, the cage containing a mouse, than the empty cage. And they spend more time actually exploring the cage with the other mouse than the empty cage. And you can see that this is not the case for the cannabis treated animals. They don't care actually whether the cage is empty or whether there is somebody in there. And the overall number of exploratory encounters with either of the boxes was also decreased in the CB1, not in the CB1, in the THC treated mice. So what does this tells us actually that the exposure actually to cannabis containing milk during this adolescent period later in adult animals produces behavioral changes that are actually relevant for schizophrenia. And that allows us now to go back and look at the brains of these animals in order to understand what is happening through this endocannabinoid exposure. Yes?
It might be that anxiety trait could explain both results. Like if they are higher in anxiety, did you check that?
Yeah, we also did. I'm not showing you here. We did a very comprehensive behavioral phenotyping of these animals, and anxiety itself doesn't seem to be very strongly affected. So we don't think that's all anxiety. But you're right, anxiety could play a role actually, especially in the exploration of the other animals. Yeah, absolutely. But we don't think that explains it. I don't think it's so important what exactly actually is going on now. It tells us that when the early exposure to THC changes how the brain responds to certain stimuli later on. And in humans, actually, we know it's associated with an increased risk of schizophrenia. So we can now try to understand in the mouse, actually, what THC is doing. And I'm just showing you very few data, how we did it. What we very often do is expression profiling studies. So what we did here is we performed an expression profiling just after the end of the feeding period and also in adult animals. And you can very clearly see this is the THC treated group. We found a number of differentially regulated transcripts. And you can clearly see that the THC-treated animals, they cluster together very clearly different from the vehicle-treated animals. In the adult animals, we see a similar picture. However, these are different genes. So we did the analysis individually for the young animals and then for the old animals. And to perhaps better illustrate this, we did a GO analysis, gene ontology term analysis. We wanted to know what is the function of the genes that have been changed by the THC treatment.
In which brain area have you checked?
This is all hippocampus.
This is after TH, young, and this is immediately after TH.
No, there is a 1 day washout period. But it was done 1 day after the last THC treatment to kill the animals and isolated RNA. And this is 4 weeks after the animals were THC free. So what this shows you also, and I'll come back to this later, is actually that THC treatment has long-term effects. We believe that in this case it's developmental effect. What we do is we change the development, the postnatal development of the brain, and that leads then to changes in the expression profile.
Cones are internalized, the rows are genes.
Yes. So each row is a transcript, a differentially regulated transcript, and each color is an animal. So we have 1, 2, 3, 4 animals here, and we have also 4 animals here, and 5 here, and 5 here. So, no, it's Illumina. It's Illumina chips. And we performed basically an ANOVA, and the cutoff rate was 1.2-fold, 5-fold induction. So, yeah.
So logically, we can say that the least effect of the oxygen, which, for example, the mother using can affect on the fetus.
No, that's something different. So the mother is exposed to cannabis. It also, I mean, this has been shown.
Now, because you said that it was after a few days washed out, you just decapitated the animal and used the brain. So if it can remain such a this long time, also we can say that for the mother and baby can be the same.
Could be the same, yeah. I believe it is. Yeah. So the message is that has long-term effects. But in this case, I said, I believe it's developmental effect. So if we perform this GO analysis, you can see that the main group of genes that has been changed is actually they are involved in metabolic processes. They respond or involved in protein tyrosine kinase activity, respond to corticosteroids, and related to the development of the parasympathetic nervous system, and so on. If we go to the Go analysis for the adult animals, it's very different actually. We have completely different group of genes here in this case. So there are many genes actually differentially regulated, related to nervous system development, to ion transport and ion channel activity, and also interestingly a very distinct group of genes related to receptor, opioid receptor binding. So that means, and this is an ongoing study that I'm showing you. This means actually that the exposure to THC will induce a transcriptional change acutely in these young animals. And these transcriptional changes then, for example, here the protein tyrosine kinase cluster of genes is one very good candidate. This differential, the induction of genes during this developmental period, can affect brain development. And that leads to altered brain function in the adult. But the next thing, what we're now doing is we're looking at individual transcripts here, and we're studying actually how they are changed during the treatment, how long they last, in which neurons are they active, and then we want to look at the fate of these neurons to see actually how they are influenced by the cannabis treatment. Okay, so this is related to the analysis of the very young mice. Now I want to show you another set of experiments that we did that are related to older animals. Because we know that THC affects cognition. If you smoke pot, actually short-term memory is affected, verbal fluency is affected, and so on. And so we wanted to know actually how the deletion of the receptor would affect cognition in animals. And what we did first is we studied young mice. And one paradigm that we used is a social recognition assay. Did you want to ask a question? No. It's a social recognition assay. Here you have two animals that don't know each other. You put them together for 5 minutes. Then you separate them, and they will explore each other. And you measure the time actually that they spend exploring each other. Then you separate them, and after an hour or so, you put them back together, and again measure how much time they spent interacting with each other. If they remember that they have previously met, they'll spend less time interacting. So, and this reduced interaction time for us is an indicator that they remember that they have previously seen each other. So if you look at young mice that are 2 months of age, you can see a normal wild-type mouse can remember the formerly seen partner around 6 to 8 hours. And that stays relatively constant during the first 5 to 6 months of their life. Young knockout mice remember the formerly seen partner much better, or much longer. So at least 15 hours actually, they remember that. But then if you look at how this curve declines actually, so they start forgetting or losing the memory for their partner when they get older, but they start losing this memory performance much quicker than wild-type mice. So this is an indication actually that the cognitive decline seems to be accelerated in CB1 knockout mice. We performed a different test called Morse water maze. You may be familiar with this. Mice have to learn how to locate a hidden platform, submerged hidden platform. And they have to use cues in the environment. So we put them in the basin. There are no cues in the basin, but they can look around in the room and orient themselves. And after a while, they will find, if we put the mouse in here, here's the platform, they will find the way to the platform very quickly. And you can measure this as a decrease in the escape latency. So they find it quicker and quicker. With more and more training, they become better and better. And in normal mice, wild-type mice, old animals, they need a little bit longer to learn this task, but there's not so much of a difference to the animals. However, if you look at the CB1 receptor knockout mice, here we have a profound difference actually. Young mice are actually doing better than wild-type controls, but as they go older, adult mice or old mice, that is completely changed. So here they really have problems in learning the task. So the cognitive decline in the absence of CB1 receptors is much faster. We look for neurons in the hippocampus of these animals, and I'm showing you only a slide here of the 3 to 5 month old animals. And you can see we have a significant loss of neurons in different regions of the hippocampus, in the CA1 region, in the CA3 region. Dental gyrus is not affected. Dental gyrus is very often resistant to these effects. We also see that we have an increased expression of GFAP. It's a normal function of aging, so the astrocyte activity increases as individuals grow older. That's also true for us. But this is much more in CB1 knockout mice. So you can quantitated this, you can see that in old mice they have a much stronger astrogliosis than the corresponding wild type in all brain regions. We also looked for the activation of microglia cells, an indicator for neuroinflammatory responses. And what we see is that the overall number of microglia cells does not change as a function of aging, but the activation profile or the morphology of the microglia change, and they have here the typical resting morphology with small cell bodies, long protrusions, and here we have amoeboid changed microglia. And again, we see a much more profound increase of activated microglia in the absence of CB1 receptor signaling. We also looked for an aging pigment,
Have you tested the glutamate level? Because we know like glutamate is highly related with the development of neuroinflammation and the effect of microglia in aging and so forth.
Glutamate levels really don't change, I think, very much during aging. We haven't actually looked at that in CB1 knockouts. No.
And did you check also neurogenesis in the hippocampus?
Neurogenesis, yes, we also looked for that. I may have a slide on this. Let's go through and see if I have it. So next we did was here lipofuxin. Lipofuxin is an aging pigment. Basically our neurons through autophagy actually they degrade inactive organelles, but highly cross-linked proteins are difficult to remove by the cells. And they form an intracellular granulate. It's called lipofuxin. Basically highly cross-linked proteins and lipid aggregates, and they show typical autofluorescence, and lipofuxin has been termed aging pigment because it increases in aging. You may be able to see this also here. In the old wild type animals it increases, but it increases much more in the CB1 mice. No, I don't have neurogenesis. We also look for neurogenesis. It's affected in young mice. Young CB1 knockout mice show reduced neurogenesis, but then as the animals grow older, so already 5 months of age, neurogenesis actually declines during aging. And we don't see a statistically significant effect in 5 months. So we don't believe that the loss of neurons can be accounted for just by reduced neurogenesis, because it's increasing at a time when we don't see a difference between the genotypes. So then we ask the question, is it now CB1 receptors on glutamatergic neurons or on GABAergic neurons? That's important. My bet was, and that relates to your question, it's CB1 receptors on glutamate neurons. Yeah, wrong. So if you look here, actually these are the glutamate-specific knockouts on the left side, and the GABA-specific neurons are on the right side. What I expected, you probably too, was that we see an aging effect in the glutamate neurons. No, absolutely no difference, but we see an aging effect in the GABA-specific neurons. So it's CB1 on GABA. That's important here. This shows you neuronal loss. This shows you astrogliosis. So the number of neurons is lower. Astrocytes go up. We also have the number of activated microglia cells increases in the CB1 receptor in the GABA-specific knockout mice. Not in the glutamate knockout mice, who have also cytokines, here also cytokines. If you look, for example, now we have the GABAs here, the glutamates on top, the GABAs on the bottom, no difference in pro-inflammatory cytokines in the glutamate-specific knockout, specific increase in pro-inflammatory IL-6 and also TNF-alpha in the GABA-specific CB1 knockouts. Yes, sir.
Impressive. What's the relative expression level distribution in glutamate? Like, so if you look at the CB1 receptor and the glutamate knockout versus the GABA, what is the whole brain in
Yeah.
CT look like?
It's present in both. It's probably more present actually in GABAergic neurons, but it's present in both.
Are there differences in regions? Like, is there one region completely missing?
No, this is looking. You mean?
Oh, this is just hippocampal, or it's whole brain glutamate knockout?
This is analysis of hippocampus. I'm only showing you the hippocampus data. So of course we also asked whether cognitive functions are specifically affected in glutamate versus GABA, and I think I only have the GABA data here. Yeah, this is 2-month-old GABAergic CB1 knockout mice. Now the knockouts still perform a little better actually in the acquisition phase and in the reversal phase than the wild type animals, the young mice, but the difference was not so striking actually. Still in the 5 months. Wait a minute. Ah, yeah. No, I'm sorry, I mixed up the colors. I can't see this very well. So in the 2-month-old mice, actually, they didn't perform better. The GABA knockout mice, they didn't perform better than the control mice already. In fact, actually, they were a little slower than the wild type mice. This became more pronounced when they were 5-month-old and really different when they were 12-month-old. So in short, we feel that the GABA specific knockouts that don't reflect the entire defect that we see in the complete knockout, but many of the age-related changes are reflected there in the GABA knockout mice. So we feel that they are more important. So, but, yeah? No.
Are these experiments done in both gender or just special gender? For example, male or female?
No, we do it in both genders. No, we actually have always the same number of males and females in all our experiments. Yeah. So what I wanted to do is actually convince you that CB1 knockouts, they show all hallmarks of accelerated brain aging. So they have an accelerated cognitive decline, accelerated neuronal loss, they have an increased brain inflammation, that increased astrogliosis, increased lipofuscin accumulation. Haven't shown you that these mice also, when they become older, develop autophagy deficits. And these changes are typically considered as hallmark changes of aging, and they're all accelerated in the absence of CB1 receptors. Now the question is this specific for the brain, or do we also find effects in that? Yeah.
So that means that CB1 signaling is actually protected in hippocampus, but CB1 signaling in cortical, probably because you've shown that in young animals, if you treat THC to young animals, it's probably induced deficits like schizophrenia. So it's like a differential.
Yeah.
It's good in hippocampus.
Yeah, but this
but
didn't.
it's not good in.
No, I wouldn't say this. We only looked in hippocampus here. So this only shows you the effect in hippocampus, but if we look at cortex, could be very similar. For
But
some,
that means that it's protective. CB1 signaling is protective over aging.
it would be protective over aging, but in young animals, too much is not good.
So we should smoke.
We'll come to this in a moment. No, seriously, we'll come to this in a moment. So this shows you, we very comprehensively looked at all organs in these mice. The only other tissue where we found in CB1 knockout mice also very characteristic changes during aging is in the skin. So typically as we age, our subdermal fat layer, whatever this is, gets thinner. So our skin gets thinner as we grow older. And that is much more pronounced in the CB1 mice. All the other organs seemed not to be affected by in terms of an accelerated age. Now if we look at the other knockouts, the CB2 knockout mice, they also have an age-related defect. They have osteoporosis. They get a much more severe age-related osteoporosis as wild-type mice. And actually I talked to colleagues, they think that the metabolic effects of CB2 signaling can also be interpreted as a form of accelerated aging. So we believe actually that in the absence of CB1 signaling, those organs where CB1 signaling really physiologically is very important, they age faster. So that leads us to the interesting question whether CB1 signaling is protective, and we studied that. So what we wanted to do is, okay, if you delete CB1 receptors, maybe smoking is good for older guys. So this is the question that we addressed in mice. We implanted osmotic mini pumps in the animals for 4 days, not 4 days, 4 weeks. Or the osmotic mini pump stayed in the animals, but they released THC only for 4 weeks. Then they had a week washout period, and then we did behavioral testing. And we did this with young animals, 2-month-old mice, and we did it with older animals, 12-month-old mice. So 12 months is pretty old for a mouse, but not super old. We can get up to 3 years in older. They released a very low dose of THC, just 3 milligrams per kilogram per day, which is so low that we do not see any behavioral effects. So the animals that we perform our tetrate of assays, we cannot detect the activity. And we performed the Morris water maze first. Now these are the results. When we do this with young animals, we see a small but significant detrimental effect in young mice. So if we elevate CB1 signaling in the animals, it has negative cognitive effects, even this low dose of cannabis. In the acquisition phase, not in the reversal phase, but also we performed the working memory Morris water maze test, also in the working memory performance that we see in impairment. So what happens to the old mice? The opposite. Now in old mice actually, CB1 signaling or enhancement by THC improved the acquisition, so they learned the task faster, they did better in the reversal phase in the task, and they also performed much better in the working memory test. So this has completely opposite effect in the two age groups. Now we wanted to see how soon we see an improvement of cognitive function. And to do this, we used a novel object location recognition test. And basically, we have a series of objects, and then we move one object to a different position, and we just see whether the animals learns actually that the position of an object has changed. And we can see this by testing whether the time that the animals spend with this object is increased. Now, if we do this, show you only the 12-month-old mice. They're pretty dumb in this test, actually, wild-type animals. So they don't see an increased exploration of the object that has been moved. However, if they have been treated with THC before, now they're much smarter, and we find an increased exploration of the object that has been moved. We find it already after 14 days of THC treatment. It lasts through the entire treatment period, which is terminal stopped here. And basically we observed them up to day 56 after that we killed the animals, and it lasted basically for the entire observation period. So the chronic low-dose THC treatment in the old animals had really beneficial, long-lasting effects. So what's going on?
So you know that during the treatment you already had this improvement.
Yeah.
So it's the same question that you did before. You know that 3 milligram per kilogram during the whole day, that represents 0.2 more or less per hour. That is an anxiolytic effect that produces anxiolytic effect of THC. So the effect that you observe during the chronic treatment, do you think that the anxiolytic effect of THC could probably improve the performance in this test?
That is a good question. So can I rule out that anxiolytic effects during these 2 days contribute? No, I cannot.
Yeah, because you
Not with this, yeah.
0. The dose that we use for anxiolytic effect is 0.3,
Yeah.
so more or less the one that you are using there.
Yeah. But that cannot, I mean, in day 42 and 56, I have no anxiolytic effects.
For sure, for sure. But I'm asking during the treatment.
No, you're right. We have to look for anxiolytic effects actually of this dose of cannabis. You're right. Yeah. No, I cannot exclude this. But we wanted to know what's going on with the brains of these mice. So the first thing that we looked for was synaptic markers. And we show you here synapsin staining in the hippocampus. You can see that synapsin staining is a presynaptic marker that declines actually as the animals grow older, so they make less synapsin. And in THC-treated mice, we found basically a rescue of this loss of synapsin expression. So if you quantitate it, actually we get more synapsin immunostaining in the brains of these animals. So this is all semi-quantitative, it's all immunostaining. So we performed also Western blot analysis, which is more quantitative. And here it's very clear actually that synapsin expression declined in older mice, and it was rescued or increased by THC treatment in the old markers. Same was seen with synaptophysin, another presynaptic markers. Again, this protein is known to decrease actually in older animals, and the level of expression was increased in the old animals. We saw a similar effect for PSD95, but here the effect of THC was very small. So we looked actually for a number of other proteins. Among those is BDNF, known to increase neurogenesis, plays an important role in synaptogenesis also. And again, the BDNF expression was increased by THC treatment. But to do this in a more systematic manner, and actually I'm supposed to ask you to cut off now the recording.